in here? No? Okay, well, I would have done them justice, but all the rest of you get to suffer. All right, for those of you who are interested, this is in Idaho, believe it or not. It's an hour north, it's in Rexburg. It's the BYU Idaho greenhouse. It's gorgeous, 80 degrees year round. Highly recommend it. They have internet access too. Do your homework in the jungle. A bit of background for plant viruses. They've been around for a really long time, obviously, and humans have only recently started recognizing them. In the 1600s, there was this big tulip mania in the Netherlands, and of course the Netherlands is famous for their tulips, but there was this particular variety, a couple of varieties, but this one was called the Semper Augustus tulip. They didn't know it at the time, but it was infected by a virus called tulip breaking virus, and that's what gives it that pattern. And it was so heavily sought after that people were paying 4,200 guilders for one flower, which translated to modern money is about $400,000 for one bulb. They are now all extinct because the virus killed them all off. After a couple of generations, they became less and less viable, and so now all we have is paintings of them. But $400,000 for one flower? Really, people? Based on their the standard wage for a middle class workman, it was about 14 years worth of salary for one flower. In 1892, the tobacco mosaic virus, which is one of the type species for plant viruses, became the first virus discovered. Like before an animal virus, this was the first one that they found. It was what they coined the term virus for, because somebody passed a liquid of this tobacco mosaic disease. They pureed up the tobacco plant and ran it through a filter that was supposed to have small enough pores to stop any bacteria. And then they took the stuff that came through the filter and put it back on a plant, and it got sick again. So then they were able to determine that it was something smaller than a bacteria that was actually causing this disease. And after several years, they were able to diagnose it as this is a virus. So virus is actually some sort, the word virus actually is a derivative of a liquid disease causing agent, because they realized it was something in the liquid that was being passed. I don't know how they got virus out of liquid disease causing agent, but somebody got creative. Tobacco mosaic virus was also the first virus to be crystallized and its structure determined. That was in the 1930s. I don't know why people were still doing science in the 1930s, but they were. And it was also used to verify the universality of the genetic code, like the codons. They used it to prove that it worked in viruses as well as in higher cells. So plant viruses have been used to do a lot of research, particularly this tobacco mosaic virus, and a lot of plant virus research that has been done on tobacco mosaic virus. So you're going to be seeing it a lot today. As a bit of background, there are thousands of plant virus species that have been discovered in the last century. Plant viruses collectively cause about $60 billion worth of crop losses worldwide every year, so they're kind of a big deal. Though most people don't usually hear about them. They hear about fungal things like blight and things like that, but you rarely hear about the plant viruses. It's possible that most plant viruses haven't even been discovered yet, because they've found several that don't even cause disease or don't seem to have symptoms, and a few that even seem to benefit their plants. In the wild, they've found some viruses that actually confer hardiness traits to plants, and they can actually modify the plants to make them repel insect vectors and things like that. So a bug that would traditionally bite this plant and feed off of the plant, the virus can actually make the plant less desirable to the bug, and hence can protect the plant, which is something that's up and coming. Kind of wild. Questions so far? All right. Now let's get into the more fun stuff. The majority of plant viruses are rod-shaped, and I don't know why that's the case. It's possible that their capsids are easier to assemble within plant cells, but I'm not entirely sure. This is an electron micrograph of tobacco mosaic virus, our friend of the day. So notice they've got these rod-shaped virions, and there are still some of our icosahedral viruses. We will run into several of those. But for the most part, they are the rod-shaped ones. Single-stranded RNA viruses are by far the most common. There are 75% of discovered plant viruses are RNA ones, and 65% of them are plus-stranded ones. So the vast majority of them, and oddly, there are very, very, very few double-stranded DNA plant viruses. Like, they are almost non-existent. It's almost entirely RNA viruses, because the next, another 15% of that is double-stranded RNA viruses. So DNA plant viruses are very rare. Mostly they are RNA viruses. And a bit of that will make sense here in a bit when we talk about why or how plant viruses are transmitted, because they have to, the way that they have to work inside the plant cells makes it a little difficult to be anything but an RNA virus. Also, and we're going to come back to this, most plant viruses lack spike proteins. If you remember, the spike proteins are the little protein that's on the outside of the plant or of the virus that binds to a host cell receptor and allows the virus to be internalized. The problem with that plant viruses have to overcome, and I think I've got the slide. Yeah. Plant cells are encased in a thick armored plant cell wall, which makes it impossible to get to receptors and be internalized. So plant viruses can't enter cells in the traditional way that viruses usually do. And so you don't need a spike protein because that method doesn't work anyway. We'll talk about later why a few plant viruses do have spike proteins. It's not for the plant. So we have to somehow overcome the armored cell walls of the plants. So that's why spike proteins won't work. So we're going to rely on various mechanical vectors for entry into the cell. They're going to use something else to physically get them inside the cell. There are a number of ways that they do this. Seed transmission is how you can pass it from one plant to another. And it's not super common. Maybe about 40% of viruses are capable of doing it. But basically, you infect the seed, and hence you infect the progeny. The tricky part is, well, how did we get into the plant in the first place, right? So there are a couple of ways that they can do that. Vegetative propagation, for those who aren't plant people, is there are some plants where you can break off a piece of the plant and it can grow into a whole new plant. Potatoes are like this. You can chop a potato up into pieces and plant the pieces and they grow into whole new potatoes. So if the virus was in the piece, then you can chop it up and you pass it on to the new guy. So this is how it can be propagated. A lot of potato diseases like potato virus Y, which is part of the potty-virus family. Isn't that an unfortunate name? So that can be spread by seed potatoes. So they have to keep an eye out for those potato viruses when propagating them, because you want to get that out of your seed stock fast, otherwise you'll spread it. Grafting, of course, with like apple trees and things like that. You can take a branch off of one tree and stick it onto another tree, and it can grow as part of that tree. If the virus was in the branch, you now put it into the new host tree. And then there's dissemination by mechanical means. There's a number of ways you can do that. Herbivores, like when you got your cows or something eating your grass, if you got an infected plant, the cow bites the grass, gets it on its teeth, and then he goes bites another grass, and ta-da, you just put it into that plant as well. You can have a contaminated knife or pruning shears. So if you clip off a branch or something from a plant that is virus infected, and then you go clip a non-infected plant, you just now infected the plant. And again, cutting the seed potatoes. If you cut a bad seed potato and then cut a healthy one, you now put it into the healthy seed potato. So you have to make sure that you're using sterilized equipment. And natural plant-to-plant contact. If you got plants that are too close, they can bump into each other and transmit it sap to sap or anything like that. They can actually just give it to each other, though that one's relatively rare because it's rare for them to be close to other members of their own species. Then the last one here on the list is actually the most important, and we're going to go into that one a bit more. These are the insect vectors, because if you notice, all of the rest of these require it to already be in the plant. So this is how you get from one plant to another. This is the main method that allows you to transmit from one species to another, or sorry, from one individual plant to another. So insects transmit about or greater than 70% of known plant viruses. So this is the main transmission mechanism. Major insect vectors include leafhoppers. I had to go look up what a leafhopper looks like. This is a leafhopper. Sometimes they're green, sometimes they're brown. They're nasty, apparently. They go and bite your plants and suck their guts out. Aphids, of course. Here's our little aphid friend up here, and whiteflies, which I'd never heard of before, but they kind of look like a moth, and apparently they're a big deal in California. Here's the general overall process about how a bug can infect a new plant. If you'll notice here, on the little aphid, it's got this little needle-like mouthpiece. And the way these bugs feed on your plants is they poke their little mouthpiece into the plant, stick their proboscis in there, and then start sucking out the contents of the plant. So here's the general process of how it works. You got the insect carries the virus, and there's a couple of ways it can do this. We'll talk about those on the next slide. But your insect carries the virus, it pokes its mouth into the plant, drops off the virus while feeding, and now the virus infects that plant, and then later on, another bug can come on, bite the plant, pick up the virus, and carry it to the next plant. So that's why these things can be a big problem. And we'll talk about later, because of aphids, Hawaii nearly lost its entire papaya crop in the 80s. Like it almost wiped out the whole industry because these aphids were spreading a virus called papaya ring spot virus from farm to farm to farm to farm and nearly took it all out. So that can be a big issue because aphids can be blown in the wind. Who knew? So the aphid doesn't have to walk across the ground. He just has to get caught in the wind and blow to the next tree. And poof, you're doomed. Questions so far? Yes. Great question. Not for the most part. And we're going to talk about here on the next slide that there's a couple of different ways that the virus can interact with the insect. Most plant viruses don't actually replicate within the bug. Like you can get them on the bug and then the bug just kind of contaminates the next guy kind of thing. But there are a couple of times where it can replicate inside the bug, and we'll talk about how that works. But as far as I know, it's not detrimental to the bug, because if it's detrimental to the bug, it doesn't get passed on to the next plant. So they're actually, they're usually symptomless within the bug, and then just hurt the plants. Great question. Yes. Oh, great. Okay. Well, then let's talk about how that works. So there are a couple of different types of insect transmission. There's non-persistent transmission, and this is simply where the bug bites the plant and gets it on its mouth, and then goes and bites the next plant and drops it off of its mouth. It's not even getting into the bug cells. It's not getting into the bug at all. It's just getting on its mouth and getting dropped off. Semi-persistent, and this one I've been struggling to try and determine what the difference is between semi-persistent and persistent. Near as I can tell, the semi-persistent means that the bugs get it into their foregut, which is this early part of their stomach right here. It doesn't actually get elsewhere into the bug. It just gets in there. But the tricky part about that one is with this non-persistent version, the bug gets it on his mouth, bites the next plant, and most of it gets off of his mouth. So then when he bites a third plant, he's not going to get that plant sick. This one, when he gets it into his mouth, he'll be able to infect several plants because it can keep getting regurgitated up with his saliva. And so it's semi-persistent. It lasts longer in the bug and allows him to infect more plants. But again, it's not actually replicating in the bug. It's just lasting longer in the bug. Here comes the persistent ones, and this is where it answers you guys' questions. The virus actually gets internalized by the bug, and there are a couple of types of persistent, because we're scientists, we have to break everything down, right? So there's circulative ones, and these ones don't replicate in the bug, but they do get eaten. And what's shown in this picture here from the textbook is circulative persistent. So this one actually gets taken up by the bug, gets all the way into its stomach, crosses the epithelium, gets into their version of a bloodstream. It's called the hemocell, and then gets transported up to the salivary gland where it can then get spit out into the next plant. So this one is persistent because these viruses are all throughout this thing, so it's going to last a really long time. So in some cases, these viruses can actually last the entire insect's life. And so that bug, once the virus gets into that bug, it will infect every plant that it bites. Propagative versions, these ones actually can replicate inside the bug. These are the ones that will have spike proteins, but the spike proteins are not for the plant, they're for the bug. So I was reading an interesting paper that was pointing out, because remember, we talked about at the beginning, these viruses have an extremely wide host range. We talked about earlier that you can have maybe 1 or 2 species with animal viruses that can get infected. You have zoonotic versions that bounce over from from animals to humans and etc. But plant viruses have an extremely wide host range from maybe 6 species to over 100 of different plant species, and they can be super wide. Like tobacco mosaic virus, if I remember correctly, can infect plants as various as tobacco, watermelons, and orchids. Like these basically, if the virus can get inside the plant cell, usually it can replicate. The limiting factor is your vector. So any plant that the bug bites, it can get transmitted to. So if you're relying on a bug that only feeds off of certain species, then those are the species you're going to get into. And because most plants have very similar replication strategies. I saw a hand over here.
Yes, you said about how viruses can jump from animals to humans. Can plant viruses jump from plants to humans? Has there been any record cases?
There was one case where somebody came up with very sketchy data, apparently, recently, where they said that there was a plant virus that potentially could get somebody sick, but it's not very well founded, particularly because most plant viruses don't have receptors. So it would have to not receptors, spike proteins. So it would have to, in order to get inside your cells, it would have to have a spike protein of some sort, and so it would have to mutate from a bug one to yours if it did do that. But that I don't know. Maybe it does happen and we just don't get symptoms similar to it being in the insect. I don't know. Great question. You have a question?
These viruses, are they vector non-specific, or are they specific vector to get to transmit them?
They're specific vector in many cases. I mean, I imagine that the non-persistent ones don't really care. But persistent ones would have to, because the virus would have to be very specific for how do I get across this bug's particular epithelium? How do I replicate inside this bug's cells? So they've found that these viruses get very, very specific, particularly the persistent versions, get very specific to a certain insect vector, and that's actually their limiting factor. So most viruses evolve and change towards the host. These viruses evolve and change towards the vector, because the host doesn't really matter, the vector does, which makes them different from animal viruses in that respect. Great question. Other questions? Okay. So virus replication, we're not going to go super in detail to how it, like all of the molecular machinery for how it replicates, because these are plus-stranded viruses. They use a lot of the similar mechanisms that we've learned about for the last several weeks. They've got hairpin loops. Some of them have 5' caps and poly A tails, etc. So we're not going to go into that detail. However, there are a couple of interesting details about how this virus does it spatially and temporally within the plant cell. And this one's been boggling my mind how they go about this. So far in the class, we've learned about these viral replication complexes where they build themselves a little cup inside the cell where they can replicate everything inside that little cup and get out. Most of those were just protein cups that they built. These guys actually build them out of host membranes. So the viruses get inside the plant, and we don't know how they uncoat yet. Because remember, before they would get internalized by the cell, and then the acidity of the lysosomes or whatnot would cause them to uncoat. These guys don't get inside the lysosome. The plant bites the thing, drops it off inside the cell. How does it uncoat? We don't know yet. We're still figuring that one out. In fact, I think they don't even know how it associates with plant replication proteins either. They just know that it gets into the plant cell and then poof, it's magically replicating. And we're still trying to figure that one out. Part of the problem is people are more interested in investing in human virus research than plant virus research. So we know an awful lot more about human viruses than we do plant viruses. What they have found out, though, is, and this is an electron micrograph, it's colorized, of course, is these plant viruses can bind to host organelle membranes, and each virus targets a different organelle. Some of them can target membranes from multiple organelles. The most common one is the endoplasmic reticulum. In this picture, the little purple thing, that is a mitochondria, apparently. And so the viruses, they go and bind on the outside, and then they start creating what are called viral replication complexes, and they can actually invaginate the membrane outward and form themselves a little bubble, a vesicle. And that's what this little white thing is. And it's in this picture, it's filled with virions, but the viral replication complex is this little bubble, and it's got a hole that you can't really see in this picture, but there's a hole that allows things to get in and out of this little bubble. And all of the viral replication machinery is contained within or on this bubble. And that's going to become important later on. Because most animal viruses, when they build these viral replication complexes, they stick to the wall and stay there. These guys are mobile. They travel throughout the plant. The whole bubble travels throughout the plant, which is fascinating and very unique. These guys can hijack host cell transport proteins. So if you guys have taken cell biology, you'll know about dyneins and kinesins. And for you medical people, myosin. These are proteins that can walk, and they travel along the microtubules and the filaments. These viruses have the ability to produce proteins that allow them to attach themselves to those walking proteins, and the plant cell will actually walk the entire vesicle with viruses still replicating on the inside in some cases across the plant towards the membrane. Because remember, we just got inside one plant cell, but if I'm going to infect the rest of this plant cell, I have to somehow get to the next cell in line. But all plant cells are covered in armored cell walls. So the bug got us in here originally, but how the blazes do I get into the next cell? Because I got to infect more than just one cell in this plant for it to be productive. So here's how you spread from cell to cell. Turns out plant cells are connected to each other. They have these little tubes called plasmodesmata. They're similar to your gap junctions in animal cells for all you medical people. But basically they're these tubes, so they all have connections, one cytoplasm to the next. So these little tubes travel through the cell walls and connect one cell to the next. So all cells in the plant are actually interconnected with each other. The virus can hijack this. So in this little picture here, we've got the virus binding to the ER here, and it's budding off to make its little vesicles here. Now, some of the vesicles get transported up near the nucleus, and they complex with chloroplasts and fellow replication complexes, and they're still determining why they do that and why the chloroplasts are involved. My guess would be it would help provide more energy, because the chloroplasts are the photosynthetic ones, so they would provide the sugar, they would provide energy for these things to replicate, but that's just me guessing. I have no idea. A couple of the rest of them bind onto these transport proteins and get walked along the microtubules and the intermediate filaments towards the plasmodesmata. Now, the problem they have to overcome is plasmodesmata have a fixed diameter. And if you're not small enough to fit through the circle, then you don't go through. And these vesicles are too big. So the virus has to find a way to get past this. So they create these proteins called movement proteins, and they're represented by little tiny triangles in this picture. But all plant viruses produce these movement proteins. And what they do is they get transported to the plasmodesmata. And a paper I saw says they're very efficient. They get to like all of them. And they bind to these plasmodesmata and physically make the pore bigger. So they actually alter the plant's plasmodesmata to make them physically wider so that the virus's vesicle can be transported through the plasmodesmata into the neighboring cell. Isn't that bizarre? And they travel outward in like a bullseye-like wave. So you've got your first infected cell, and then it hits the movement proteins, get all the plasmodesmata on that cell. The virus gets transported out to neighboring cells, and then the movement protein transports out to those ones' plasmodesmata and infects outwards. So they've actually done experiments where they used fluorescent proteins to track these movement proteins, and they actually go out in a ring. And all of the plasmodesmata that are outside the ring are normal sized, and all the ones on the inside are big. Isn't that cool? And the infection ability of these viruses is extremely efficient. You only need 1 to 15 copies of mRNA in a cell to establish an infection. It's that good, which is why these replication complexes are capable of pulling this off. Part of the reason why it can get away with that is because you're taking the whole bundle with you. So you've got the ability already to pump out lots of viruses. You can avoid the plant's intracellular defenses because you've already got the bubble going. So once you're established in one cell, you're home free. You can get into any cell you want. Questions so far? Okay. Now, here's our problem, because diffusing across these membranes takes a little bit of time. And somebody in 1935 did this very historical experiment. If we were relying on the plasmodesmata to get through every single cell in the plant, it would take us all year, right? This virus needs to get through this plant a little faster than that. So they did this little test to figure out how the virus transports through the plant. So this is a tomato plant, which can be infected by tobacco mosaic virus, our old friend. So they took a tomato plant that was about medium sized, and they inoculated one leaf, which is represented by this little striped leaf right here. They inoculated one leaf with tobacco mosaic virus. And then over time, because they did this to several tomato plants, after each day they would take a couple of the tomato plants and slice them up. And then they would allow each of the slices to incubate to grow up any residual virus that was in it. And then they would test to see which pieces of the plant had the virus in them. And that way they could track over time without having fancy technology like electron micrographs and fluorescence and whatnot. They could track where the viruses had traveled throughout the plant. And this is the story that they put together. It takes about 3 days. I don't know where my mouse is. There it is. It takes about 3 days for it to travel from plasmodesmata to plasmodesmata throughout the leaf, to kind of expand throughout the leaf. For those 3 days, only that one leaf was infected. Then on day 4, it hit the phloem. Now, for those of you that it's been a while since 101, plants have things called xylem and phloem, which is essentially their little bloodstream. So the xylem pulls water from the roots and sends it out throughout the plant. Then the phloem takes the sugars that are produced during photosynthesis and transports them throughout the plant. So these are physical tubes that are at the center of the shafts of the plant and the veins of the leaf that allow them to transport resources throughout the plant. So once the virus is able to hit those after 3 days in the leaf, it can travel relatively quickly throughout the plant. So on day 4, it hit that and traveled all the way down to the roots, oddly, and not anywhere else, just the roots. So it shoots down through the stem and goes down through the phloem down to the roots. Then on day 5, it takes off and goes back up to the top of the plant to infect young leaves specifically. So they found that the older leaves, they dodged those and went straight for the younger leaves first. Maybe that's because they're replicating faster. would be my guess. Though the interesting thing is I saw a paper that said that mesenchymal cells, which are their version of a stem cell, the kinds that are rapidly dividing, they dodged those completely. So it's got to be a somewhat differentiated cell that's still replicating faster. And I don't know why. And I'm not sure if the scientists know why either, because remember, we need money, right? So if you guys ever become multi-billionaires, invest in plants, right? They are the future. So then after about 10 days, it started getting to some of the more medium-aged leaves. And then, but notice, this one was in 1-day intervals up here. It was shooting fast. Down here, we're down to 10 days. That's a 5-day jump. So it took it a really long time to get to the medium and older leaves. And oddly, if they infected it in a very young tomato plant, the younger leaves never became, the older leaves never became infected at all. The virus just completely avoided them. And I don't know why. I couldn't find that answer anywhere. This is a really old experiment, and I don't know if they had the ability to figure out why. But I thought that was particularly interesting. But by using this method, and this is again with this particular virus, some viruses travel faster depending on the plant and the virus. Some of them do it slower. Some of them, it takes them only a day to get out of the leaf and things like that. But if you'll notice over time, within a couple of days, you got the whole plant. So if the next bug comes and bites anywhere on that plant, it can transmit it. And because of those plasmodesmata, you can actually transmit from one cell to the other before you even have virions packaged, because all you have to do is move the mRNA. So you don't have to fully infect one cell and lyse it to move to the next one like some viruses do with the animal cells. You simply just have to get in that one cell and then transport the mRNAs from one cell to the next. So they can travel a lot faster that way. Questions so far? Didn't lose anybody on the ancient image that I photographed out of a book? Okay. So this is a bit of heading into where the future of our study of plant viruses are. Like we said, plant viruses cost us a lot of money. And we mentioned earlier that in the 1980s and 90s, a virus called papaya ring spot virus nearly took out Hawaii's entire papaya industry. And this was a big deal because apparently the papaya industry, who knew, is a $100 billion industry worldwide. So there's a lot of money in papayas, in case you're looking for a farming degree, right? There's a lot of money in papayas. And Hawaii relies heavily on their papaya industry. But this virus, it's transmitted by aphids. It's blown from tree to tree. And so the aphid bites the tree, it infects it. There's no treatment for it, as is the case for most plant viruses. Basically, you have to try and catch it before it spreads throughout your plant. And hopefully you catch it before an aphid bites the thing and moves it to another tree. So the only treatment is chop down your tree and pray the aphid didn't buy it before you got to it. So they were wiping out tree after tree, and the virus can reside in the soil. So if you have a virus infected area, the only mechanism of avoiding that is pack up and move to a new farm. So the virus slowly conquered the Hawaiian Islands until they narrowed it down to one valley. I think it was on Oahu, and that was the entire papaya industry was in one valley. And then in 95, it broke into the valley and started getting transferred there. So they were about to lose everything. And in one of the cool stories of GMOs, because most GMOs are developed by private companies like Monsanto, and you have to pay a lot of money to get it. In this case, the University of Hawaii decided they were going to try and save their state. And so they developed a GMO papaya plant that was resistant to the papaya ring spot virus and gave away the seeds for free. And by doing so, they saved the Hawaiian papaya industry. So about 76% of Hawaiian papaya is now this new GMO rainbow papaya. So if you've ever eaten papaya here, you've probably had it. It's the resistant version. Interestingly, it's only resistant to the Hawaiian strain of the papaya ring spot virus. So the version that's in Guam, if it ever gets to Hawaii, they're in trouble again and they're going to have to start all over. But so far, it's doing good. And it's 20 some odd years later. I think they started selling it in 96 or 98. So it's about as old as we are. They've also done this with a couple of them. There's I know of one called plum pox virus. They genetically modified a plum to be resistant to plum pox. Isn't that so fun? You like kind of want to have plum pox. You're like, I don't know, maybe they're like these little, maybe you grow plums. You're like, that you can also breed virus resistance because some of these plants have natural genes that can help protect them. Sometimes you got a version of a protein that, because these viruses have to hijack host cell proteins sometimes in order to work. So maybe it develops a version that can't work as well with the virus. And so then it gains a natural genetic modification that protects it from these viruses. And so plant breeders can actually breed varieties that are more resistant to plants or to plant viruses. But again, it's a really big problem worldwide. They've lost wheat crops. There's apparently a big epidemic down in Africa of cassava, which we don't really eat here, but it's a major crop. It's like wheat for us. They eat cassava. And so by wiping out the cassava, they're having a lot of problems down there in Africa. And so they've got to try and figure out a way around that one. So this is kind of the future of how to control these. I mean, you can spray for bugs and whatnot and try and keep the bugs off. There are planting strategies where you can plant certain plants that will actually repel the bugs or attract bugs to those plants. It's kind of like a bait plant. So you can plant the plants around your field or intersperse them with the crop so that the bugs will go to those plants and not to your actual crops, and that can help protect them as well. But this is kind of the genetic future for they're trying to develop these virus-resistant varieties. Any questions? All right. Well, sorry we flew through. That's all I have. So thank you for coming.
Super cool.
Isn't that kind of fun?
Why is Lane so afraid of these?
I know, right? It's so funny. I was talking to Dr. What's his name? Reinhardt. I think he's on he's in the Gale building, but apparently he's the plant expert here on campus. I was talking to him and told him that I was teaching Dr. Lane's plant lecture, and he's like, she doesn't want to talk about plants. Well, I'm her faculty mentor, so she's gonna hear about that. I was dying.